So now moving on to Figure 2. Figure 2a describes the overall layout of experiments to be discussed in the following slides. Again, it is about direct reprogramming fibroblasts into induced neurons. However, scientists are specifically looking at investigating the transcriptional dynamics during reprogramming to identify the pathways that orchestrate induced neuronal conversion. In order to do this, scientists used RNA sequencing data from bulk fibroblasts as well as cells undergoing conversion at 1, 2, 3, 16, 18, and 24 days. As a refresher, RNA sequencing is a recent transcriptomic approach where the total complement of RNAs from a given sample is isolated and sequenced using high-throughput technologies. So they found that there were no systematic efforts that had been made to utilize broad and unbiased datasets to identify pathways and corresponding modulators that orchestrate the induced neuronal conversion. Therefore, they wanted to test this experiment out. So, using the Ingenuity Pathway Analysis software tool for RNA sequencing, scientists discovered more than 500 pathways called to be significantly transcriptionally enriched or depleted as a conversion of fibroblasts to induced neurons proceeded. Figure 2b shows a selected 10 enriched pathways that scientists within this study wanted to further examine. Specifically, it shows the activation z-score of the indicated signaling pathway on the left-hand side. Z-scores represent a statistical measure of the match between expected relationship direction and observed gene expression of a given pathway. Typically, z-scores greater than 2 are considered significant within this experiment. The bottom axis refers to the timeline described in the overall experimental layout in Figure 1a. It indicates day 1, 3, 16, 12, 18, and 24 days on induced neuronal conversion. As you can visually see, as the days go on, there is more significant z-scores greater than 2 because there are more transition to blue. However, scientists wanted to focus on the most promising signaling pathways. These pathways are described in Figure 2c. So within each of the pathways, scientists discovered small molecules that act as activators or inhibitors in the pathway to increase or decrease the conversion of fibroblasts to induced neurons. Therefore, scientists wanted to discover which combination of these small molecules would promote the highest conversion to induced neurons. So how did the authors decide which of the 10 signaling pathways and small molecules were the most promising and which ones to use? For this, we need to discuss supplementary Figure 1 for Figure 2. In this figure, scientists tested various small molecules with fibroblasts to see the expression of PSA and CAM-positive cells. The dashed line on the graph indicates the NC control. Anything below this line had low expression, which correlates to low efficiency of converting fibroblasts to induced neurons. Anything above the line in green shows high expression. Due to this, scientists took 4 small molecules above the dashed line to create a combination concoction. An important thing to note on this graph is the zero scores. Zero scores represent the molecules that were lethal to direct reprogramming, which is why scientists ruled these molecules out. From this, the 4 small molecules chosen were pentigrin, which is an integrin activator, AZ960, which is a JAK2 inhibitor, CM336372, which is a RAF1 activator, and KC7F2, which is an HIF1A inhibitor. The combination of all 4 small molecules are now known as Z-Pak, and this combination will be further described in the next coming figures. As I've stated before, scientists have now created a combination of 4 small molecules with the hypothesis that it will increase the expression and conversion of induced neurons. And this particular combination is called Z-Pak. In Figure 2d, immunocytal co-staining of neuronal markers beta-3 tubulin were performed. On the left-hand column, the only medium added to cells were the control NC medium. On the right-hand column, the medium NC plus the Z-Pak combination cocktail were added to the cells and then stained. Visually, you can see that there is higher expression of beta-3 and NEUN neuronal markers than that within the NC medium alone. This tells us that the Z-Pak combination increases direct reprogramming. So specifically looking at figures 2E to 2H, the induced neurons derived in the NC plus any of the four compounds resulted in a significant increase in the number of NEUN and beta-3 tubulin positive cells compared to those derived in the NC medium alone. This quantitatively exhibits the increased efficiency of Z-Pak, and this confirms the scientists' hypothesis that Z-Pak increases the number of fibroblasts converted into induced neurons. Now moving on to the third experiment of this paper. Figure 3A describes the overall layout of the following figures. This has to do with investigating the changes induced on the transcriptional level by the Z-Pak cocktail. In order to properly do this, scientists within this article performed time series RNA sequencing from fibroblasts from 3 donors that were converted to induced neurons in either an NC medium alone or NC medium plus the Z-Pak cocktail for 5, 10, 15, and 20 days. So in total, 3 biological replicates from 4 time points resulted in 24 samples to experiment with. So, figure 3b compares the top 100 upregulated and top 100 downregulated genes for the conversion process for both conditions. From this, scientists were able to detect an 80% and 93% overlap respectively. Figure 3b also shows a heat map of proteins that were upregulated or downregulated, where orange equals low expression and blue correlates to high expression. In this, scientists found that there was a high similarity of expression on day 5, 10, and 15. However, there were more significant differences on day 20. So scientists believed and hypothesized that a difference in composition of neurons at day 20 differs from those at day 5, 10, or 15. Therefore, further experimentation needed to be explored and is exemplified in the following figures. This slide will discuss 3 figures for the third experiment. To begin, let us discuss figure 3C. This figure shows a comparison of FPKM normalized gene counts for the conversion process of both NC and NC plus Z-Pak conditions at day 5, 10, 15, and 20, because scientists hypothesized that at day 20 there is a difference in the neuronal expression. Here, scientists were able to detect that the transcriptomes induced by NC or NC plus Z-Pak were actually highly correlated, with a Pearson correlation coefficient of greater than or equal to 0.85 for all sample time points. This indicates that there's actually a broad transcriptional similarity between the induced neuronal process in NC and NC plus Z-Pak cocktail. This directly leads into figure 3D. This figure shows a heat map showing relative expression of the glutamatergic neuronal specific genes. Therefore, the hypothesis that there is a difference in composition of neurons at day 20 from day 5, 10, or 15 is false, due to figure 3C and 3D. It actually shows that there is a glutamateric similarity across them. Lastly, in figure 3E, it shows a hierarchical clustering demonstrated a clear separation of NC and NC plus Z-Pak groups except for 2 NK samples at the earliest point of conversion. So rather than differences lying in latter days at day 20, there's actually more differences at earlier points of conversions at day 5 or earlier. So, the last figure in figure 3 is figure 3H. This shows a rank hypogeometric overlap, or RHO map, that compares the gene expression differences between NC and NC plus Z-Pak cocktail to expression differences between matched fibroblasts and the adult human brain. To give you a rundown on what the map tells us, each pixel represents an overlap between the two conditions to fibroblast or adult brain transcription, color-coded according to the negative log value. On the map, the extent of shared genes upregulated in NC plus Z-Pak cocktail and the adult brain is displayed in the top right-hand corner, whereas the shared genes upregulated in the NC, medium alone, and fibroblasts are displayed in the bottom left-hand corner. Scientists observed a pronounced bias in the overlap of the Z-Pak cocktail with the adult brain, indicating that the Z-Pak cocktail-induced neurons were indeed more similar to brain expression patterns than previously thought, and it perchance has to do with the maturity of the fibroblasts. Before I move on to the final figure, a little background information needs to be done on methylation. So, DNA methylation is a process by which methyl groups are added to the DNA molecule. This is also an epigenetic mechanism used by cells to control gene expression. Methylation can change the activity of a DNA segment without changing the sequence. When located in a gene promoter, DNA methylation typically acts to repress gene transcription. This background leads us directly into Figure 5. And yes, we did skip over Figure 4, but that is because it has more to do with the pathways, and a further detailed analysis on those specific pathways would be required to have any useful discussion on it. So, Figure 5 aims to compare young, middle-aged, and old fibroblasts to see if they can retain their epigenetic age. As I've described in previous slides in the beginning of the article presentation, retaining the epigenetic age is important in determining and studying age-related disorders. So to move on to Figure 5a. Figure 5a shows a clear difference between young and old donors, with middle-aged donors showing an intermediate age-dependent CpG methylation pattern. Methylation patterns of the young and old induced neurons by the Z-Pak cocktail were largely unchanged compared to their parental fibroblasts. So to give you a more rundown on this actual figure, high methylation levels are shown in yellow and low methylation levels are shown in blue. So the methylation of one young and one old fibroblasts are paired, and unconverted fibroblasts are shown next to its induced neuron counterpart to show the comparison between the two. So in Figure 5b, it wanted to see whether or not the fibroblasts maintain their epigenetic age. And to apportion the majority of the variation, principal component analysis based on all 850,000 CpG methylations consistently clearly separated the samples with respect to their age as the largest component. Therefore, it can be concluded that direct reprogramming and with the Z-Pak cocktail, the epigenetic age of fibroblasts and induced neurons can be maintained for them to study age-related disorders. In a rough conclusion, the experiments performed in this study showed that you can increase the efficiency and accuracy of direct reprogramming of fibroblasts into induced neurons. At the beginning of this presentation, I mentioned that the study could be used to test age-related disorders. This is due to the unique ability of direct reprogramming to maintain epigenetic age of fibroblasts in cells. However, there were no experiments performed in this study that directly looked at any age-related disorders. However, I believe before that you can test age-related disorders, my hypothesis first needs to be tested. I believe that it is important to investigate how well aged fibroblasts perform with direct reprogramming. My hypothesis for an additional experiment is fibroblasts from aged individuals are less likely to reprogram to induce neurons than those from younger individuals, and I will discuss this further and the experimental techniques used in the next slides.